Hello students, today we'll discuss microbiology syllabus for the 5th semester. In your 5th semester, there are 2 papers which you have to write for microbiology. One is applied microbiology and the second one is immunology. Today we'll discuss the immunology paper syllabus. In this paper, you are having 4 units. The first unit is history of immunology and immunity. Here we'll basically learn how exactly immunology originated, what were the first discoveries, who are the scientists who made some breakthrough discoveries which actually led to the development of immunology. And we'll also learn what is immunity, the concept of immunity in the human body, what are the different types of immunity, and what are the components of this immunological system. We will learn about antigens, what components can act as antigens, and what are the characteristics of antigens, and what are antibodies, and the different types of antibodies and their structure and properties. We'll also learn about complement system and how the complement system gets activated. So once you have an idea about the different components like the antigen, antibodies, complement systems, we can move on to learn about the immune system, the components of the immune system. So as you know, any system in the human body, it is made up of cells which aggregate to form tissues and then an organ system. So immune system is one of the organ systems. So first we have to learn the cells that are present in the immune system, which is basically your blood cells, right? So we will learn all the different types of cells that are present, right? What is their function, how do they mature, how do they develop, we will learn about that. And then we'll also learn about the organs, what organs actually constitute the immune system, right? Which are these organs, what is their structure, their anatomy and their roles, their functions, right? So then we will learn one of the important parts of immunology, which is actually having lot of application in diagnostic labs today, that is the antigen and antibody reactions, right? So we will learn how an antigen and antibody react, what are the types of reactions, and we will learn that how we can use these reactions in diagnostic techniques, right? So we will learn different techniques, right, which can be used in diagnostic laboratories, right? So some of the techniques will be ELISA, RIA and immunofluorescence. Then we'll also learn some of the other processes that are involved in immunology, concepts like hypersensitivity, autoimmunity. And we'll also learn about the production of monoclonal and polyclonal antibodies, right? So antibody plasma therapy is having a lot of application these days. So we will learn how these antibodies can actually be produced in the lab. And of course the most important topic in this paper will be vaccines, right? As we all know, vaccines are actually a boon to the mankind, which has saved us from so many diseases. So we will learn how these vaccines are made, what are the different types of vaccines that are present, and what are the different diseases for which vaccines have already been made, right? So we will discuss all these topics. So this is what we will be discussing in your immunology paper, and with this you will learn about how a body fights against microorganisms, pathogens, foreign bodies, what helps the body in providing resistance, and how we can deal with it, and also a lot of applications of how we will use this in diagnostic procedures, right, in performing tests.